Okay, so now we're going to focus on the first stage of individual ID annotation. So just to reiterate, what we've done is to click the launch button and then selected the individual ID tab. So basically, in terms of the settings here, we're just going to leave the default batch size as usual. We can then select the species of interest. In this case, we're going to be working with leopards for two reasons. Firstly, because Hotspotter works really well on them, and also secondly, um, there's a nice number of images for demonstration purposes. Next, you can select your stage of individual identification. Since we haven't performed the first stage, we can't move on to the second stage. So we have no choice but to start at the first stage, which is why we can't actually select anything here. We're forced to perform the first stage first, and that is what's called cluster identification. And I'll go into exactly what that means again shortly. Next, we can select what algorithm we want to use. And at this stage, we've got two options. So we've got Hotspotter in conjunction with our heuristic algorithm, or the heuristic only in a case where Hotspotter doesn't perform well on any particular species of interest and is just going to get in the way. In this case, we're going to use Hotspotter, so we do know it does work well on leopards. So what Hotspotter is it's a coat pattern recognition algorithm that's open source and is essentially the industry standard at this stage. And what that does is it, like fingerprint recognition, gives us a similarity score between two images of a leopard based on how similar their coat patterns are. And so it works really well on highly patterned species, so stuff like leopards or zebras or giraffes or anything like that. So that's what we're going to use here. In terms of the heuristic, basically that looks at a couple different things. Primarily, it looks at what we call a space-time map of an individual, which sounds fancy, but essentially it's just a spatial map that's also a function of time. And essentially the core concept is there if two individuals or two animals have been detected at the same site within a close amount of time, they're more likely to be a potential match and should be boosted in terms of similarity. Or, if they're at one site over in the same evening, then they're also more likely to be the same individual. So that's part of it. And then the other part of it is informational tags. So you'll see now when you launch this task, you can set up informational tags for your individual. So they're fully customizable. So you can do stuff like male, female, adult, juvenile, but also a lot of sort of more custom stuff like scar, missing ear, missing leg, missing tail, snare injury, anything like that. That's sort of any identifying information. And so then again, the idea is two individuals with a more overlapping set of informational tags are more likely to be the same individual and hence should be seen sooner in that, boosted in terms of their similarity. So that's the idea there. So then to just swing back around to what cluster identification is, we did cover it already, but just to reiterate, the idea is that when an individual, a group of individuals walked in front of your cameras, plural, at a site, multiple images were triggered, and we've hopefully got now multiple view angles, and hopefully in particular multiple flanks of each individual. And so what we want to do then is look at each cluster at a time and be able to use the contextual information of as the animals progress chronologically along to be able to combine, be able to say this individual in this image here is the same as this individual in this other image here, and then we can combine those view angles to improve AI performance going forward. And as I say, in particular, trying to combine multiple flanks. Beyond that, we're also trying to, at the same time, to audit our AI-generated flanks. So we have an AI that looks at every single detected animal and tries to predict what flank we're looking at from the animal's perspective. That's quite important, so just keep that in mind. So the left flank is the animal's left flank. And obviously the right flank is its right from its perspective. And so that is automatically predicted, but obviously it's not perfect. So just while you're performing this task, you're also just double checking that those flanks have been detected correctly and correcting them so that we can just calculate similarity between all the lefts and between all the rights individually. You also see that there's a third class called ambiguous, where you can't, it's not really either. And essentially then quite loosely, we just combine left and ambiguous, right and ambiguous, and work with it that way. Um, One last thing that's also worth pointing out is that even though this is called cluster identification, and I'm talking about being delivered one cluster at a time, what we actually also do is cluster the clusters, which sounds like a mouthful, but essentially the idea is any clusters of your species of interest, i.e. leopard in this case, that were triggered at the same site within half an hour of each other are delivered together in this annotation interface, because often if when you're looking at sort of rarer species like this and you get a redetection at the same site within that short space of time, it's often the same individual, and then you can try and attempt with contextual information to be able to determine if it's the same one and try and get multiple flanks of that individual. So you'll see that there, that's all within the interface. So now we can start. So essentially what we can do is click launch now that we've set up our various settings. Now we can set up our informational tags. I'm not really going to be using these, so I don't claim to be an expert at identifying the sex of a leopard, but I just want to show that for demonstration purposes. So in this case, we've got manual, um, male and female, and M for male, F for female. As I say, you can add a whole lot more if you want, and you can submit that. And now you'll see that the data set will go into a processing state. This may take some time depending on how large your data set is. What we're essentially doing at this stage is using our AI to try and predict what flanks what flank of each animal is visible in each image, so that you can now correct it. So we'll continue once this completes. And there we go. It's now finished launching, so we can go to the jobs page and grab a job. So yeah, so this is what the interface looks like. We've got two panels of images. So we've got one on the left, one on the right. And what you'll note is that both sides actually have the same set of images, the exact same set of images. So if I page to them simultaneously, you'll see that they're all the same. And so the idea is that we're looking at one cluster of images, and that same cluster is shown on both panels at the same time, to allow you to be able to look at any possible combination of images within that set. Some other things just quickly worth pointing out is that these two images, okay, that's the same image. Well, no, it's not, sorry. So these two images are from the same camera, which you can see is indicated by a green camera symbol. And obviously visually we can see that. But if we zoom in, we can look and we can see there's a camera trap just past the animal over there. So if I actually scroll to the left, we can see now the camera symbol is switched to red, indicating we're looking through two different cameras. And that's basically what we can see is these two images are 4 seconds apart. And that pretty much indicates that's exactly what we expect. We can see the leopard investigating the camera over there, and from the side we can now see whether in the camera that's the recipient of that interest. So that's sort of the basic idea there, so that you can now sort of contextually figure out which images belong to which individual. So obviously with leopards being a much more solitary species, it's mostly going to be one individual, and you need to manually check that they are indeed the same individual, because obviously you don't want to mistakenly combine individuals. So it's worth checking and just making sure you do that correctly. But obviously, if something for like spotted hyenas, something like that, we've got a whole pack of hyenas walking through. It's a bit more effort to be able to actually say, okay, this hyena over here is that one over there. But yeah, and that's what leopards make for a nice example here. So the first thing that we also want to do as well is, while we're at it, is we're needing to check our flank information. So we can see that this is indeed, remember from the animal's perspective, it's the animal's right-hand flank. So that's correct. Indeed, still their right-hand flank, and we can see from context images from the same camera at the same timestamp they're the same animal. So I can left click on that box there, and then left click on that right box there, and I'm saying that one over there is the same as this one over here, and they get combined. So you'll see it automatically generates a name. In this case, individual number one. You can edit that if you want. You can add some extra notes if you want. And then you can add your informational tags. In this case, I'm not sure, so I'm going to refrain from adding a tag, and I'm simply going to submit. You can see now the color of the box has changed to indicate that these are now the same color and hence the same individual. And then if I mouse over the box, I can see which individual it is. It's individual number one. So that's now those two, that's the same image again. So now we can flip to the opposing camera, and we can say, okay, well, these two images are 4 seconds apart. I mean, if you want to take it very seriously, you can look at the spots and confirm that 100% they are the same for those two different flanks. So you probably can't do that in this case, but we're quite confident they're the same. So I'm going to left click that box there, and then left click that box there to say that one is the same as that one. And that's the core idea of what we're trying to do here. And again, you can see now it's also changed to green, and it's individual number one. And same with this box over here. So we're going to say that one over there is the same as that one over there. And once you've then associated all those boxes or all detected animals in a cluster with an individual, the platform will automatically take you to the next cluster. So you don't need to do anything. It'll just say, okay, you're clearly done, and it'll move you on. If you do realize that you've made a mistake, when you've done that, you can simply click. You can see there's the back option with the same tilde hotkey we always use. If you press that, it'll take you back to the previous cluster, and then you can make any changes you want. We'll go through that shortly. But essentially, you'll see now there's a new option as well to now submit. So basically, going forward, it's easy for us to tell when you're done with the cluster, but when you're editing, we don't know how many changes you're wanting to make. So rather than have it be annoying and kind of keep going forward every time you make a change, you just simply say submit when you're done, and it'll bring you back forward again. So in this case here, I can immediately see that we've got a cluster of clusters. So remember I said that we actually cluster our clusters on a half-an-hour interval for extra information. And so if I can see that because there is a boundary between these sets of images. So these two, you can see are 0 seconds apart. So at the same timestamp, so presumably the same individual. You can see the flanks, left and left, that's correct. And then I switch over to the next image, the other side of that orange boundary, and we can immediately see now that this cluster symbol has turned red, indicating to us we are now in a different cluster. That's just to make that quite easy. So what we can do now is see that that's a minute apart, minute and 6 seconds, and you may or may not remember that clusters are defined based on a 1 minute time difference. So that's why these two have been split up. So basically, what also worth pointing out here is you can see that this has been detected as a right flank, which is not correct. This is a left flank, but I mean, it's not really of value as a left flank because you can't actually see the left flank. So what you can do is now right click on that box, and then you can relabel it as whatever you want. In this case, I'm going to actually just say ambiguous because it's not really a good example of a left-hand flank. We can carry on then. The other image, taking the same timestamp, left flank, that's great. We can see there's another boundary, so we can go into even another cluster. And now we can see we are 15 minutes apart. And so again, that's why we use the 30-minute barrier. We can now try and determine whether this individual is the same one. And it's quite likely it is. And I think it actually is. We'll just double check. Obviously you can page the rest of the images, the right flank, that's all correct. This one, I don't know if you can even call what you would even call that, and it's not immediately obvious. So again, I'm just going to actually label that as ambiguous in this case. And then we've got left-hand flank and left-hand flank. So what we can do is basically then say, okay, well, let's use these two left-hand flanks, 16 minutes apart, to see if they're the same individual. And they indeed are. So that's lovely. So you can see there's these 4 rosettes here, and with this sort of shape around it. So there's your 4 rosettes and that same shape. So we can now see that they are indeed the same individual. So we can say that one over there It's the same as that one over there. And again, it auto-populates the name, and you can fill in your other characteristics and hit submit. So what I can then do is say, okay, well, we've now identified that those two as being the same. So I can then go right back to those two images and say, well, um, those two at the exact same time instant. I can double check the spots if I want to, but they're the same. So that one over there is the same as that one over there. So now we can start, we can move on to this intermediate cluster and say, okay, well, I can already see it, but I'll show you an image just for sanity's sake. There's those four rosettes with that same pattern around it. So it's all one single individual. So we click that one over there, it's the same as that one over there, and now its color changes as well. This one is now from the third cluster. So even though that's now a right flank, we can see that it was taken 33 seconds. So it's up to you whether you want to combine those two, but they're the same. In this case, it's good enough for our purposes. So what I can do then is say, okay, well, that one is still the same as this one. And then those two are the same. And then I think we're just missing okay, I'll store these two. So there we go. Those two are the same. And those two are the same. Okay, I'm still missing one apparently. There we go. And there we go. So we've now associated all boxes with an individual in the cluster, and you can immediately take them to the next one. So here we go. That is not a right-hand flank. Straight off the bat, that is a left-hand flank, remember, from the perspective of the animal. Then we look at the next image. That is a left flank, 8 seconds apart. That's the same individual, right-hand flank, left-hand flank, and left-hand flank. Again, you can see now it's a cluster of clusters. We're talking 1 minute apart, but we can see it's all the same individual. So now I can show you one of the shortcuts, so we don't have to do the whole thing we just did for the last one, which is really useful for solitary species like leopards, and that's the single individual shortcut. So what that does is says there is one single individual in this entire cluster of clusters. And so we just press the hotkey M or press that button, click that button, and it says, okay, what do you want to name your individual and whatever is usual. In this case, it's probably a large male. So I'm going to select male and then submit that. And that's it. And it says all those detections are one individual, nice and easy. So what's worth pointing out though with that as well is that if there are, you'll only be allowed to do that if there's only a single detection in each image. If there's multiple detections, then it can't be a single individual, so you won't be allowed to use that shortcut. So just quickly cover some of the other options we have there. Undo just undoes your last action. So let's say, okay, I've combined these two, that individual and this individual, and it's now individual number 4. And I say, oh wait, no, that's a mistake. I can then press U or click this button, and it undoes that last action. Nice and simple. Delete deletes an individual. So again, I'm going to combine them again. They're now individual 5 now. And what I can do then is delete an individual. So if I press delete, now I'm in delete mode. You can see by that button changing to a cancel button, which obviously you can click cancel to just exit the mode. And then you just click on the individual you want to delete. So what it does is it just dissolves that individual. So all the detections that are associated with it, it just removes, it makes that individual, it dissolves it. Other option is to center back. The best way to describe that, so again, it goes into center back mode, and then you click on the detection you want to change. And the best way to describe that is to sort of equate it to like Microsoft Word or something like that, where you have two objects or two images on top of each other, and you can't click on the one that's behind the one in front. So what you do then is say center back, and you click the box in front, and that gets sent to the back so that you can now click on the one that was behind it. You should quite rarely need that, but sometimes it's necessary. So that's what that's there for. Now, this is a nice example. We also have the option here called unidentifiable. And what that does is allow us to mark a detections, like in this case here, is unidentifiable, meaning you'll never be able to identify that detection with an individual. You could combine it. This is all going to probably be a single individual again, and you could associate it with that individual. It doesn't really make a big difference, but also in general, it's better to just say, well, that's not really of use. So it's up to you. In this case, you probably just include it with the individual, but you can say unidentifiable, in unidentifiable mode, click on that individual, and have a mouse over it, now it says unidentifiable. And what it does is basically you'll never see that detection, that image doesn't go anywhere, we still have it, but you won't see it again in the individual ID workflow because you won't be able to combine it with anything. So it's for cases where it's too blurry, the animal's wiped out by the flash, or obscured by foliage or something like that. Back I've already showed you, and some individual I've already showed you. So that's it. So we've gone through the full functionality. So now we can just finish off some clusters and see if we get some nice examples. So basically now we just need to decide whether these two are the same individual, and I'm going to say they're the same. So right flank, left flank, that's probably ambiguous as well, I guess. And then we can say that one over there is the same as that one over there, and submit. Nice and simple. Here we've got two images from the same camera, from the same timestamp, nice and easy. So I just simply press N as the shortcut for single individual, and then I can press Enter to submit that form. So that becomes the shortcut for leopards a lot of the time, which is nice. So again, these two are the same. Left flank, right flank, it's correct. It's single individual, hit Enter. Right flank, left flank, those two are the same individual from the same timestamp essentially. Well, different timestamp, but same moment in time. You can see the flash of the cameras there. And then we've got clusters here from 1 minute apart, and we seem to have a female and a male. So those are two different leopards. So this is exactly why we check this stuff. So we can now say, well, these two are the same. So that one over there is the same as this one over there. I can hit my hotkey M to make it a male, enter to submit. And then now this is something else that's worth showing, is there's only one image, one detection of this individual. So the question then becomes, well, how do we get a single image or single detection individual? So it's actually quite easy. We simply say these two panels on the exact same image. I click on the box there, and the same box over there, and now that's individual number 10. Really straightforward. Here we've got presumably a single individual again. So I'm going to use the shortcut, left flank. Ooh, that flank is incorrect. Right flank. And enter, and we submit. So now we've got that leopard, left flank, that leopard, right flank, and that is now the left. That's right, but kind of ambiguous again. So I'm just going to go ahead and assume this is a single individual because this is just for demonstration purposes. I don't want to spend too much time on this. New single individual submit, left flank, right flank, single individual, and submit. In this case, this is actually a nice example of where you want to use the unidentifiable option. You'll never associate that with anything. I'm not even entirely sure if that is a leopard. It might be something else. But the point is now we can just say unidentifiable, and since it's only a single detection, it'll apply to that detection. You don't have to even click on the detection. Here we've got the same individual, so I can then say single individual, male, submit. Here's another interesting example. So we actually have two animals in the same image. It's going to be not really any different, but it's worth just pointing out. So I've got now left flank, that's correct. You can see the collar over there. So I can immediately say, I don't know if you can see it, but I can see it, that animal's got a collar. So I can say that one over there, it's the same as this one over there. Their flanks are correct, and submit. Then I've got one more image over here. Yeah, that one doesn't have anything. So that's a right flank, and if I zoom in, no collar. So I'm going to just go ahead and assume that these two are the same. Flanks are correct. I can combine them. Nice and simple. Left flank, single individual. And there we go, we're done. So as usual, you just get a pop-up when you're done, asking you if you'd like to go back and make any changes, or submit and continue. So I'm going to say we're done. And there we go. We should be finished now. Just wait for the page to update. And so you'll see that the task will go into a wrapping up state. And again, this may take some time depending on how large your data set is. Um, so basically, it's at this point that we actually run all the images through Hotspotter in this case, now that we know the flank information and stuff like that. So it may take some time depending on how large your data set is.